Alright, hello. So, I would like to introduce the cell cycle. The cell cycle is a complex set of stages which is highly regulated. It has checkpoints which direct it and make sure it is on the right track before moving on to the next stage. Alright, so the cell cycle, like any other cycle, it loops continuously. And it's divided into 4 different sections. The first being G1. The G in G1 stands for growth, which is it's an area or a period of time where the cell simply grows. The next is S, which stands for synthesis. This is when DNA is copied, and DNA is copied. After that comes G2, which is more growth along with preparation for division. So this is when the cell is getting ready to divide. And after that comes mitosis. Mitosis, as you probably all know, is prophase, metaphase, anaphase, and telophase. And this stage also includes cytokinesis. Running out of room. And cytokinesis is basically just separation of the cytoplasm. So there are 3 checkpoints along the cell cycle. The first being at the end of G1, the second at the end of G2, and third and final one at the end of mitosis. So these checkpoints at these stages make sure that everything is fine before moving on. So these are particularly useful for stopping cancerous cells from dividing. And these checkpoints work using chemical signals, both internal and external. Some examples of these would be MPF, which stands for mitosis promoting factor, and it would be located right here because it's promoting mitosis. And another example would be PDGF, which stands for platelet derived growth factor. Alright, so this chart shows how cyclin, it basically controls the entire cell cycle. So a cyclin is basically just a protein that's associated with cell division. And yeah, it controls the cell cycle from G1 all the way to mitosis. Okay, so here we have a cancer cell. And it wants to go through mitosis and replicate. So it starts going through the cell cycle. But there are checkpoints which essentially stop it from getting to replication. So it tries to replicate, but it stops at the first checkpoint, which eventually leads to it dying off. However, if these checkpoints aren't working properly, like if a protein isn't functioning as it should, then it can easily travel through. And then it can replicate, which at that point it becomes a serious problem. It'll start multiplying first to 2, then to 4, then to 8, and you get the rest. And I hope this helped.